Alrighty. Need to close the door. Give me a second. Give me a second. Alright. So, in this lecture, we're going to talk about tissue engineering, which is a favourite topic of mine and something I find very fascinating in that the very specific ways in which cells interact with their environment affects a whole lot of stuff in terms of their internal chemistry. So, thus far, we've only talked about how growth factors and the internal chemistry of cells determine the cell behaviour. But that's a really kind of narrow look at the way cells grow. Because in fact, the way that cells grow in tissue and in the body are really different. And indeed, the different matrices, the different scaffolds or structures that the cells are growing in, has a really big effect on the way that they grow. So, for example, if we're trying to culture, say, muscle fibers in tissue, now they're nice and adherent and they like to adhere to the surface. But once they've proliferated and covered the entire plate, they're going to stop growing, and they can't grow in three-dimensional space. They can't grow in a third dimension because there's no structure for them to grow in that direction. Now, of course, in your muscles, there are three dimensions, right? It's a three-dimensional shape. So just from that alone, the cells that you culture in your two-dimensional culture are going to be different to three-dimensional culture. And that's the point of tissue culture, is that we need to try and make a structure for our cells to grow in that is as close as possible to the true physical environment, so the structural and connecting environment that they have in the body. And that's what we're going to talk about today. So, how complicated is tissue engineering? Might just go through a little structure. So we're going to talk about the complexity of tissue engineering and some of the different tissue engineering proteins that go into it. Am I in focus? And in addition, we're going to talk about the way that cells sense the extracellular matrix, that's what we call it, how they emit and interact with the extracellular matrix, the fact that there are growth factors in the extracellular matrix, so the ECM is complex. Collagen is complex. Transition factors? I don't think it's transition factors, it's growth factors. Signals are in the ECM, right? Cells and their with ECM, and then a couple of the scaffolds that we use. Now, through a lot of this, it's kind of very example-based. It's very like, oh, look at this example of something, and I'm not really that prone to giving you all your information through examples, because in your own research, you're going to need to know a whole lot of detail about a few things. So I'll talk about maybe one or two examples, but if you want to research fibronectin, go nuts. But I'm not going to tell you everything you're going to need to know about anything. So why would I tell you not enough about everything? So let's get into it. So there are 25 different isoforms of collagen, right? 25 different isoforms of collagen. Can't even see that, that's terrible. 25 different isoforms of collagen. And so what that means is that even amongst the most basic of our extracellular matrix proteins, there's a whole lot of different functions, okay? There are connecting ones, there are elastic ones, there are kind of long, strong ones, which is the A series and the A10 series. A1 series? Yeah. The A1 series are responsible for connective, and A10 series are responsible for binding cells together. And that's kind of the important thing that we need to know about that, is that there's a lot of proteins that work in the extracellular matrix, and that makes it very complicated. The structure that each of our cells is interacting with is very, very complicated. In addition, there's agarine, right? There's also some laminin, there's also some elastin, laminin, and proteoglycans. And I'm going to talk about proteoglycans a little bit because it's a nice example of a little bit of complexity in the extracellular matrix, but by no means is this comprehensive, and by no means am I giving you enough information to really understand what's going on. So let's look at a proteoglycan. So a proteoglycan has a protein backbone, right? So that's a protein backbone. And it also has glucose aminoglycans hanging off it. Right, these are glucose aminoglycans. And the reason that's interesting is because glucose aminoglycans attract water very strongly. So we can imagine we have our little water molecules and they like to sit in here next to the GAGs, and this is abbreviated to a GAG, by the way. Some people call it a GAG. And you get your water molecules in here, and they're hanging out, and they're attracted by electrostatic forces. Now, why is that interesting? That's interesting because now we have a structure that kind of fills up with water that's been attracted by electrostatic forces, and then if you apply a lot of force to it, the water goes out, right? It's being squeezed out of the sponge, so to speak. And so where do we think that this kind of structure would be useful in the body? Well, it's useful somewhere that you need to apply a lot of force to something and get a bit of give out of it in a compressive way. So this is a compressive protein, compressive structure, and you apply force to it and all the waters run out, right? And so you get compression of this structure, but only in the places where the most force is applied, and so it's a nice cushioning protein, right? And so where would that be found in the body? It's going to be found in places that need cushioning. So in your joints. And that's just one interesting example of an extracellular matrix structure within the body. Now, what else do we have? So now we're going to talk a little bit about how do cells sense the extracellular matrix, right? And cells have things called integrins. Integrins, and this is important, right? And so we have our cell wall, and we have our integrins. And they span the cell wall, right? And you have an alpha integrin and a beta integrin. Alpha, beta, right? See that? Yep, cool. This is your cell wall. And then in here, you can cause cell signaling on the inside of the cell due to the transmembrane proteins, which are the integrins, and they receive signals from the extracellular matrix. But the important thing is that integrins are designed to bind only to very specific extracellular matrix proteins. By which I mean that, what's the importance of that? Well, if you've got say muscle cells or cells that should exist in the walls of your vascular structure, and those gain or lose these integrins, they change their integrin structure, and they're allowed to roam around in the bloodstream, then you can get unchecked proliferation of cells that really shouldn't be in the bloodstream, which is kind of a progenitor for cancer, and it's all very dangerous and it's not the best thing. So one of the big roles of these integrins is cell death, and it inhibits the cell death inhibition. So it inhibits cell death, right? So we're having something bound here in an extracellular matrix, be it collagen or be it whatever, that inhibits, in healthy cells, that inhibits death, and that's important. So that's one way that cells sense and interact with the environment, and the important thing to know there is there's a whole bunch of different integrins, heaps of them, and they always come in pairs, and those pairs allow us to interact with different kinds of extracellular matrix proteins, and they allow us to localize the cell to a specific point in the body and get information from specific proteins. And you don't want them going rogue, because then you'll get tissues in places that you don't want them. Okay, the other way that cells interact with the extracellular matrix is through kind of a weird little tensile test thing that they do. So you've got your integrin, and this is your extracellular matrix, and you've got your little cell, and the cell has actin in here, right? And that actin is pulling on the integrins, and testing how strong the extracellular matrix structure is. It's testing how strong it is. It determines just how much compressive strength the extracellular matrix has, and therefore how much tensile strength, how much tensile force we can apply to this actin. And that's important because it affects the way that specific cells differentiate. Right? So if you have a weak extracellular matrix, like this, or if you have a strong extracellular matrix, you're going to get kind of a different cell structure, and you're going to change the way that the cells divide and differentiate according to this plot that I'm about to draw, which is pretty exciting. Alright, so this is stiffness, and this is this is probability. And I wish I had multiple different pens, but no. And so we're here. 1, 2, 3. Right. And so this is 1 kilopascal. That's 10 kilopascals. That's 100 kilopascals. And this is important right now, okay? So the 10 kilopascals, the force, the stiffness that the cells are detecting in the extracellular matrix seems like very low stiffness in engineering terms. But it's important to note that that stiffness is on the molecular scale. It's the stiffness that the cell itself is detecting. And that's different to the macroscopic stiffness we might expect in engineering materials. Right. And so we have these 3 different levels of stiffness and 3 different probability curves, and this is osteo, so bone, this is muscle, and this is neuronal. Right? And these are the optimal growth stiffnesses for each of these cell types. So what we're saying here is that in order for a cell to grow optimally, you need to have the correct stiffness of the extracellular matrix. Okay, now the final thing that we're going to talk about is remodeling of the extracellular matrix. ECM remodeling. I don't mind if that's spelled wrong, whatever. Okay, so it's important for the cells to be able to interact with the ECM and remodel the structure. Say, for example, injury, or just homeostasis. Right? In a lot of situations, cells need to be able to break down the extracellular matrix and change the structure, replace wounded tissue, or break down a blood clot, create a blood clot, all of these kind of things that are involved in homeostasis involve the upkeep of the extracellular matrix and therefore the breakdown and build-up and the remodeling of the extracellular matrix. Now this is important because in the extracellular matrix, there are growth factors. In the extracellular matrix, there are growth factors. And what that means is, as the extracellular matrix gets remodeled, there can be the release of growth factors that stimulate the growth of different tissues. So, for example, if you get injured, there's a lot of FGF2 in the muscle tissue. And so as that muscle tissue gets broken down because it's injured, it stimulates the growth of new muscle tissue. And that's kind of one of the feedback mechanisms that your cells use to keep things effective and homeostatic and recovering well from injury. Okay, and then the last thing, the actual last thing, I lied the last time, is to talk about the cell matrices that we use currently. So it's important to note a couple of things. Number one, cells like to remodel ECM. Right? 2 ECM is complicated. 3 TGA approval is hard. Right? So these 3 factors combine to mean that the best and most effective way for us to create tissue engineering constructs is to use the simplest things we can. Now in the past, what was used was bioactive polymers, which had bioactive binding sites on them. But the problem with that is you'd get a form of rejection where the cell body would create this really fibrous kind of structure around the biopolymer and just bind it up in this really useless thing. It just kind of rejected it. And so we want to avoid that. And so in order to avoid that, we need to use things that the body not only can remodel but is kind of happy with. So it couldn't remodel these bioactive polymers. And so things that it can remodel are, for example, porcine extracellular matrix, which is just the matrix taken from a pig, and you decellularize it, and then you put it into your culture, and the cells go into it, and they, you know, it's very familiar. It's generated by a real organism, so it's not artificial, and it's much easier for those cells to work with. There are a couple of other structures which we can look at, but that's the general gist of the way that goes. There's also bioactive gels, there's also electrospun fibers, there's also attempts to use apples in combination with electrospun fibers. There's also, what's the other one? Aerogel, and like agarose and things like this. So the important thing to note with TGA approval is everything that we try and use in these extracellular matrix should already be medicinally approved. Messing with this stuff and also having to get approval so that your ECM can be implanted into a human is a game that we don't particularly want to play, so we try and avoid it. Okay, so that's our very quick summary of tissue engineering. So, cells like to remodel extracellular matrix. There's a whole bunch of complexity in extracellular matrices. There's 25 different kinds of collagen at least, and there's a whole bunch of other proteins in addition to that. Cells can interact with the extracellular matrix, and they like to do so. They have integrins, and they can also sense the force. And when we create our own extracellular matrices, we try and keep it as simple as possible and try and not use new materials that need to be bio-approved. Okay, and that is tissue engineering. Thanks, guys.